In this video, I'm going to discuss what I call feral tissue, which is the term I use for tissue growing in the body that you don't want. It can get larger and larger, and it can migrate to other parts of the body. First, let me give you a short introduction. It will then be followed by a very long technical explanation. But for those that want a short understanding of it, here it is. The microbiome of the gut is designed to make short-chain fatty acids. These short-chain fatty acids get into the cells through something called a hydrocarbon receptor. If the microbiome is damaged or disturbed and there are not enough short-chain fatty acids, the first thing that happens is more hydrocarbon receptors are expressed on the cell. But if that's not enough, the model I'm proposing states that the cell itself will start making short-chain fatty acids on its own. The long-term issue with this is these short-chain fatty acids increase the acidity of the cell environment, and that triggers cells to become feral. Also, with more hydrocarbon receptors, other things can get into the cells. Viruses, aflatoxins from fungi and mold, parasite metabolites, and chemical toxins can all get into the cell using the hydrocarbon receptors, and they can all accelerate the transition from normal tissue to feral cells and feral tissue. The premise is, therefore, that due to a lack of short-chain fatty acids and the presence of one or more of the following, being viruses, fungi, parasites, and/or chemicals, tissue can shift from a healthy state to a feral state. A protocol you might consider, therefore, would be to supplement with short-chain fatty acids while you're trying to get your microbiome to make it for yourself, detoxify the viral, fungal, parasitic, and chemical toxins that may have gotten into the cells across these receptors, and neutralize or buffer the acid environment that the tissue is existing in now. That's the simple version. If you want the more technical version, here it comes. Worldwide, over 27,000 people die every day from unwanted and aggressive growths of tissue inside their bodies. For the purposes of this video, I will refer to these unwanted and aggressive growths of tissue as feral tissue. I'm using the term feral tissue because the word that we normally use for these unwanted and aggressive growths has a lot of scientific and emotional baggage associated with it. It's a word that no one wants to hear from their doctor. It's a very frightening word, and the protocols associated with treating it are violent. Surgery, radiation, chemical cocktails. These protocols are based on the idea that we must kill feral cells, that we are at war with them, and that we must accept a degree of collateral damage, personal injury in the process. But in spite of over 4 million published studies and billions of dollars put into research, other than earlier diagnosis, we haven't had any major breakthroughs. If this is a war, it's a war of attrition, and we're not winning. Why present a new model? Well, it's been said if you always do what you've always done, you'll always get what you've always got. The new model I'm going to share is very different from what is classically taught, causes feral tissue. I chose the term feral tissue because I think it is the best description of what it is. If you don't feed your family dog long enough, he may turn feral and break into your chicken coop and eat a chicken. Try to take that chicken away from him and he may snarl and snap at you. He's gone feral. But put a plate of food in front of him and he may well eat that food, then lick your hand and go to sleep by your feet again like the good dog he once was. Calling unwanted and aggressive growths feral tissue reminds us that we may have unwittingly played a part in turning part of our body into this feral tissue, and that by understanding why it became feral in the first place, we may have a chance to return it to a healthy and benign state again. With all that said, let us begin. Why have we been so unsuccessful in dealing with what I call feral tissue? Why is it the number 2 cause of death, even after decades of research? I believe it is due to 2 mistakes made over 50 years ago. In 1976, researchers discovered that dioxin, the highly toxic chemical, could bind to a newly discovered receptor in our cells. The name for something that binds to a receptor is called a ligand. They called this new receptor the aryl hydrocarbon receptor. For simplicity's sake, I'll refer to it as the hydrocarbon receptor. Researchers found out that when dioxin, a toxic ligand, attaches to this hydrocarbon receptor, it causes some genes in the cell to turn on and others to turn off. And as a result of this changed gene activity, healthy tissue in our bodies can become feral and behave in aggressive and life-threatening ways. However, not everyone that develops feral tissue has exposure to environmental toxins, so it was reasoned that for some people, feral tissue is due to genetics. After all, it is changes in how the genes operate that are creating the feral tissue as a result of the toxic ligands like dioxin. This set the narrative, framed the scientific conversation for the last 50+ years. The search was on for the environmental and genetic causes of feral tissue. But calling the receptor a hydrocarbon receptor, I believe, was a mistake. The first oil well we know of, historically speaking, was dug in China in the year 347. We haven't evolved with oil long enough to have special receptors made just for hydrocarbons that oil creates. It turns out that this hydrocarbon receptor can actually bind to quite a few ligands. It can bind to certain viruses, mycotoxins from mold and fungus, parasite metabolites, and yes, certain chemicals like hydrocarbons. So, should we call it a viral receptor, or a mycotoxin receptor, or a parasite metabolite receptor? No. Just because these ligands can all bind to the receptor doesn't mean that that is the receptor's purpose. What do all these toxic ligands have in common? They all cause feral tissue. Okay, we could call it a feral tissue receptor. That would be an improvement. But is that what it is? No. What it actually is a short-chain fatty acid receptor. It is meant to bind to short-chain fatty acids, the ones that a healthy microbiome makes, namely lactic, acidic, propionic, and butyric acid. The issue is that the short-chain fatty acid receptor can also bind with other things we don't want it to bind to, and as a result, feral tissue can develop. Once I understood that the hydrocarbon receptor was actually a short-chain fatty acid receptor, a new model of feral tissue began to form in my mind. Was it the missing key to understanding feral tissue, why it forms, and how we might turn it back into normal, benign tissue again? So, what causes feral tissue? Environmental toxins can create feral tissue. This we know. But what about genetics? Researchers have identified approximately 50,000 different genetic mutations that can be found in feral tissue cells. But are these mutations causing the tissue to become feral, or is the feral tissue causing the mutations? It's an important question. We have to know cause and effect. Thanks to groundbreaking studies by Dr. Seyfried in 2010 and 2015, we now know definitively that most feral tissue is not caused by genetics. In Dr. Seyfried's studies, he inserted the DNA, the genetics, of feral tissue into a normal cell, a healthy cell. And lo and behold, that normal cell did not become feral, even though it contained the genetics of a feral cell. These experiments prove that feral tissue is not driven solely by genetics. So, what does cause feral tissue? First, a disclaimer. I'm not a doctor, and nothing I'm about to talk about should be considered medical advice. Nothing I mention is meant to treat or diagnose any condition. That being said, we have between 79 and 103 organs in the body, depending on how you define an organ. Let's be conservative and call it 79. Since I consider the microbiome, that is, the mass of bacteria in our gut, to be an organ, that gives us 80. If you haven't watched my other videos on the microbiome, please pause this video now and watch them. It will help you understand what I'm about to say in greater detail. Okay, welcome back. So now you know how important the microbiome is for our health and how common it is for it to be out of balance. One of the main functions of the microbiome is to make short-chain fatty acids, specifically lactic, acetic, butyric, and propionic acids. These short-chain fatty acids enter our bloodstream and have a profound regulatory effect on our health. Short-chain fatty acids regulate our brain, digestion, endocrine system, immune system, neurotransmitters, and so on. One reason why short-chain fatty acids have so many regulatory effects is because they activate receptors and affect enzymes all over the body. Receptors like G-protein coupling receptors and enzymes like histone deacetylases. Think of your body as a factory with 36 trillion machines, our cells, each with hundreds of different kinds of dials, which are the receptors. These dials have to be continuously adjusted to keep the system running properly. Since most of us have compromised microbiomes, this means that most of us have low levels of short-chain fatty acids, or worse, the wrong kinds of short-chain fatty acids. This means that the dials on these 36 trillion machines, the receptors on our cells, aren't being properly adjusted. We can experience this as fatigue, inflammation, blood pressure issues, brain fog, and 1,000 other disorders. Since the body needs short-chain fatty acids to operate properly, what can the body do if a person's microbiome isn't making short-chain fatty acids in adequate amounts? Well, I believe that the body adapts. I believe it finds another way to make short-chain fatty acids, a backup manufacturing process. I believe that the body creates a new organ whose job is to make the short-chain fatty acids that the microbiome isn't supplying properly. And I believe the name for this new organ is a benign growth. That is, tissue that is making short-chain fatty acids for the body, but is still stable. A benign growth is not growing rapidly, nor is it displaying the aggressive behaviors of feral tissue, such as cells migrating to distant locations. Not yet, anyway. Okay, that's quite a claim. Do I have any evidence to validate such a theory? Let's see. If the body were going to make a new organ, what tissue would it make it from? Most likely pluripotent stem cells. These cells can become anything. And yes, we now believe that many unwelcome growths in the body begin as pluripotent stem cells. What else would we expect to see if this theory were correct? We'd expect to see that these unwelcome growths were actually making short-chain fatty acids. Are they? Let's look at the 4 short-chain fatty acids one at a time: lactic, acetic, propionic, and butyric acid. First, let's look at lactic acid. I understand that technically it is not a short-chain fatty acid, but for the purposes of this video, let's consider it as a short-chain fatty acid, as it is somewhat similar. One of the first scientists to suggest that feral tissue was a metabolic disorder was Otto Warburg. He showed that in feral tissue, the Krebs cycle, the main energy-producing cycle in a healthy cell, was going backwards. A backwards Krebs cycle still makes energy, but at a 94% loss of efficiency. Surely this must be a mistake. To be so inefficient, something must have gone wrong. No sane cell would do this on purpose, right? A backwards Krebs cycle in feral tissue is inefficient, I agree. Inefficient at making energy. That would be ATP, the energy currency of the body. But it is wonderfully efficient at making the short-chain fatty acid, lactic acid. To be more technical, glucose is burned in the cell to create pyruvate in a process called glycolysis. The next step is to take the pyruvate and burn it again in the Krebs cycle in a process known as oxidative phosphorylation, or OXPHOS. However, in feral tissue, at least in the early stages, pyruvate doesn't burn again in the OXPHOS pathway. Instead, it ferments and creates lactic acid. Okay, feral tissue makes lactic acid. That's one of the 4 short-chain fatty acids. But the current model says that lactic acid in feral tissue is bad, that lactic acid causes many of the hallmarks of feral tissue behavior, such as angiogenesis, that is, the creation of new blood vessels, immune dysfunction, which would be lower T and natural killer cell activity, and also suppressed apoptosis, which is the process by which damaged cells are supposed to self-destruct. It was also believed that lactic acid contributed to the migration of cells from the feral tissue to other locations in the body. But is lactic acid really what's causing these problems? In the 2016 scientific journal Oncotarget, Doctors Wu and Ying published their study where they added 20 mm of lactic acid to feral tissue cells, but kept the pH at 6.7, which is nearly neutral, only mildly acidic. And what happened? The feral tissue cell started acting like a normal cell again. We could say it went from feral to benign. This tells us that it isn't the lactic acid per se that creates feral tissue, but rather it was the low pH that the lactic acid created, or should I say, the inability of the cell to buffer the acidity of the lactic acid it created. Here's what I believe happens. When the microbiome doesn't make enough short-chain fatty acids, such as lactic acid, tissue forms a benign mass, and this benign tissue will create lactic acid and other short-chain fatty acids for us. This is a good short-term strategy, but it can cause long-term problems. Over time, the short-chain fatty acid production creates a very acidic local environment, and I believe it is this acidity that shifts benign tissue into feral tissue. Okay, that's 1 short-chain fatty acid down. Lactic acid, 3 to go. How about acetic acid? Do feral tissue cells make acetic acid? I haven't seen any research that looks at how much acetic acid is in feral tissue, but what I have seen is that the 3 enzymes needed to make acetic acid are all very high in feral tissue. These enzymes are ATP citrate lyase, which turns citrate into acetyl-CoA, followed by histone deacetylase, which then turns acetyl-CoA into acetic acid. We also see elevated levels of acetate kinase, which makes acetic acid from the phosphorylation of acetyl phosphate. Okay, what does the current model say about feral tissue and acetic acid? The current model is confused by the fact that endogenous acetic acid, that is, that which is produced inside the cell, is vital for feral tissue cell proliferation, whereas exogenous acetic acid, that which comes from outside the cell, reduces cell proliferation. This confusing statement makes perfect sense in the model I am proposing. The issue with acetic acid on the inside of the cell is that, like lactic acid, it increases the acidity of the local environment. And remember, an acid pH is one of the main drivers of the structural and metabolic changes we see in feral tissue. The extracellular fluid around the feral cells can be as much as 63 times more acidic than normal cells. If I had to live in a vat of acid, I'd misbehave too. On the other hand, exogenous acetic acid, that is, acetic acid made in the microbiome and in the bloodstream, has a tendency to shift feral cells into a benign state. This agrees with the model's prediction, which would say that short-chain fatty acids coming from outside of the cell, that would be exogenous production in the microbiome, would tell the cell that it no longer needs to be in a feral state and can become benign. whereas short-chain fatty acids inside the cell would be formed as a result of a deficiency state outside of the cell. Okay, if this low pH is really bothering benign and feral cells, how might they adapt to this acidic pH, which is the result of the short-chain fatty acids they're tasked with making? They could do this by lowering an enzyme called sodium-potassium ATPase. And what do we find in feral tissue cells? Low levels of the enzyme sodium-potassium ATPase. And what happens as a result? A 5-fold increase in sodium levels, which I pose is being used to buffer the local environment. So there's another mystery of feral tissue solved, the mystery of the high sodium levels. Unfortunately, the same sodium-potassium ATPase enzyme that helps alkalinize the area is also responsible for cell adhesion. Cells are supposed to stick together. Prostate cells stick to prostate cells, and breast cells stick to breast cells. Cells are not supposed to wander off, to migrate, to go to distant locations in other parts of the body. Unfortunately, when the enzyme sodium-potassium ATPase decreases, cells don't stick together as well. The result is feral cells can migrate to other locations and start growing there. What else might feral cells do to neutralize their acidity? How about increase their alkaline calcium levels? Do we see elevated calcium in feral tissue? Yes, feral tissue has altered calcium channels and pumps, which results in an increase in calcium. This calcium helps buffer the acidity, but it also comes at a terrible price. Elevated calcium increases immune evasion, angiogenesis, and the migration of feral cells, to name just a few. Okay, that's lactic and acetic acids, 2 down, 2 to go. Let's look at propionic and butyric acid. The data I have for propionic and butyric acid is more circumstantial, but this may simply be due to the fact that these two short-chain fatty acids aren't as deeply researched as lactic and acetic acids in feral tissue. First, let's look at propionic acid. There is a pathway to make propionic acid in cells called amino acid catabolism, which is the breakdown of certain amino acids, one of which is methionine. If feral tissue were using methionine to make propionic acid, what would we expect to see? I would think we would see decreased levels of methionine as it was consumed to make propionic acid. We know that in later stages of feral tissue status, people lose weight. And what do we find in the very early stages of this weight loss? Methionine is the very first amino acid to disappear. Is methionine being used to make propionic acid? What about butyric acid, the fourth and final short-chain fatty acid? Do feral tissue cells make butyric acid? In normal cells, there is a process called beta oxidation, where fatty acids are broken down to make acetyl-CoA. Yes, this is the same acetyl-CoA that turns into acetic acid. We know that feral tissue cells have altered beta oxidation. Scientists report that as a result of this altered beta oxidation, fatty acids aren't completely transformed, and instead, small bits of fatty acid are left over. Small bits of fatty acid. Well, another way to say small bits is short chains. So, are the small bits of fatty acid they are finding simply short-chain fatty acids? I believe that just as the Krebs cycle goes backwards to make lactate, the beta oxidation pathway can go backwards, and instead of burning short-chain fatty acids, it can make them. I believe it is making short-chain fatty acids, and perhaps butyrate is one of them. Reversing the Krebs cycle and beta oxidation pathways are both things that bacteria can do. We know that the Krebs cycle goes in reverse in feral tissue to make lactic acid. I think it is plausible that feral tissue reverse the beta oxidation pathway to make other short-chain fatty acids as well. Alternately, beta oxidation may still be going forward, burning fatty acids, but not burning them completely, leaving short-chain fatty acids behind as a result. Well, that's all 4 of them: lactic, acetic, propionic, and butyric acid. Let's recap a bit. We know that feral tissue is not caused by genetics. There's very good evidence that feral tissue is being triggered by low short-chain fatty acid production in the microbiome. In the beginning, I mentioned this misnamed hydrocarbon receptor. How does this fit in with the model? Hydrocarbon receptors are made to bring short-chain fatty acids into the cell. And what do we find in feral tissue? A very high level of hydrocarbon receptors. They're trying to soak up every last bit of short-chain fatty acid. Unfortunately, this also means they're soaking up lots of toxic ligands like viruses, aflatoxins, parasite metabolites, and other toxic chemicals as well. What happens in the absence of short-chain fatty acids and the presence of toxic ligands is that the hydrocarbon receptors bring the toxic ligands into the cell. It's important to understand that the hydrocarbon receptors aren't like an off-on switch. They're more like dials. They have very different effects on our DNA depending on what they bind to. Once a ligand is transported into the cell by the hydrocarbon receptor, it lets go of the ligand. If it's a good short-chain fatty acid, the cell can use that short-chain fatty acid for fuel. If it's a toxic ligand, on the other hand, then one of three things can happen. The toxic ligand can reattach to the hydrocarbon receptors and the cycle repeats. The toxic ligand can bind to the DNA and damage it. Or the toxic ligand can be detoxified by the body. We want the third option. We want our cells to detoxify toxic ligands, but these are difficult toxins to get rid of. It is possible, and I'll explain in the protocol part of this video at the end how. But for now, let's get back to the hydrocarbon receptors. In an inactive state, the hydrocarbon receptors are surrounded by special proteins called chaperones. In this case, 4 heat shock proteins. When the toxic ligands bind to the hydrocarbon receptor, these 4 heat shock proteins are displaced, where they also have effects on the cells. Normally, heat shock proteins are a good thing. You get them from saunas and cold plunges, and they make you stronger and more resilient. In the case of feral tissue, they make the feral tissue stronger and more resilient, which is not necessarily what we want. Toxic ligands also get the genes to decrease adhesion molecules like integrin, which, like an acidic environment, triggers feral cells to separate from the main body of tissue and migrate through the blood vessels and lymphatic system to remote locations. Toxic ligands also can suppress the immune system so that it won't go after the feral tissue. This is why viruses, fungi, and parasites target our hydrocarbon receptors. By controlling this receptor, they can manipulate our immune system to tolerate them. Okay, let's talk a little bit more about pH. Current therapies target feral tissue cells specifically, but the feral cell itself may not be the best target. What I think we might want to do is focus more on the environment of the feral cell, and pH is a major aspect of the environment. Normal cells have an extracellular pH of 7.3 or slightly higher. Feral tissue cells have an extracellular pH of roughly 6.5 to 7.1, but sometimes as low as 5.5. At 5.5, it would be 63 times more acidic than normal. The idea that acidity makes feral cells more aggressive is not new to science. It is called the acid-mediated invasion hypothesis. One researcher went so far as to state that feral tissues are selected for their ability to export acid, meaning they're evolving to become better and better at pumping out acids. Okay, but which acids? If we replace the word acids with short-chain fatty acids, then the statement reads that feral tissues are selected for their ability to export short-chain fatty acids, and that agrees with this model. Back to pH. Once an acidic pH triggers the loss of adhesion molecules, these free-floating feral tissue cells move out of the local acidic environment. Once they migrate to a place that has normal pH, their adhesion molecule program reboots, and the feral cell then sticks to the new tissue wherever it finds itself. So, to minimize the migration of feral cells to other parts of the body, it is imperative that we neutralize the acids in the feral tissue extracellular fluids. I will discuss how this can be accomplished in the protocol section shortly. Looking at feral tissue as a response to and a replacement of a failed microbiome, can help us make sense of the many otherwise confusing aspects of feral tissue. The microbiome should make short-chain fatty acids. The feral tissue, I believe, makes short-chain fatty acids. As a result of the acidic pH in the intestinal environment due to short-chain fatty acid of the bacteria, intestinal cells slough off in the feces every few days. As a result of the acidic pH in the environment of feral tissue, feral cells can slough off into the bloodstream and lymphatics and migrate to other parts of the body. Bacteria in our microbiome have two growth speeds, which is controlled by something called the acetate switch. Acetate is a version of acetic acid, a short-chain fatty acid. When bacteria grow slowly, they eat acetic acid. But when bacteria shift into their fast-growing state, they create acetic acid. This is the acetate or acetic acid switch. When normal tissue is growing slowly, it also eats short-chain fatty acids. When feral tissue grows quickly, it creates short-chain fatty acids. Our own tissue seems to have an acetate or acetic acid switch, or should I say a short-chain fatty acid switch. Bacteria in the microbiome communicate to one another and coordinate their actions. Feral cells that have migrated to other locations in the body also communicate to one another to coordinate their actions. Let us recap. So, what causes feral tissue? I think it is a combination of several events. 1. Insufficient short-chain fatty acids from a compromised microbiome triggers the body to create benign tissue growths which are tasked with making short-chain fatty acids. 2. Over time, the localized acidic environment resulting from the short-chain fatty acid production in these benign growths triggers certain metabolic and structural changes that leads to what I call feral tissue and the migration of feral cells throughout the body. 3. The absence of short-chain fatty acids leaves the hydrocarbon receptors open to attaching to viruses, mold toxins, parasite metabolites, and toxic chemicals, which, along with the increasing acidity at a certain tipping point, turns the benign growths into feral tissue. Here is where I believe genetics finally comes into play. How much resistance we have to this, that is, how far along we can go before we reach our own personal tipping point, and where, and what kind of feral tissue we may develop, that, I believe, is affected by our genetics. One test of whether a new theory is superior to a current theory is whether it can explain things that the current theory cannot. So, let's look at some mysteries that this new model may explain that the current theory cannot. 1. Why do feral cells have more hydrocarbon receptors than normal tissue? The increase in hydrocarbon receptors is a result of low short-chain fatty acids, because the more hydrocarbon receptors a cell has, the easier it is for that cell to capture the few short-chain fatty acids that may be available to it. 2. Why are feral tissue cells intermixed with normal cells? Feral tissue cells feed on the carbon dioxide which is released by normal cells, and in turn the normal cells get short-chain fatty acids from the feral tissue cells. It is a symbiotic relationship. 3. Why do feral tissues that have migrated to various parts of the body communicate between one another and transfer elements between them? Feral tissue is trying to maintain short-chain fatty acid levels. By communicating with other growths of feral tissue, they are all better able to coordinate their efforts. 4. Why does surgically removing or debulking the main body of feral tissue often trigger the accelerated growth of any remaining but smaller outposts of migrated feral tissue? Once the main body of feral tissue is removed, the level of short-chain fatty acids drop again, and this triggers the remote feral tissue outposts to grow faster to make up for the lowered short-chain fatty acid production. 5. Why does carbon dioxide stimulate feral tissue? Carbon dioxide both lowers pH and feeds the reverse Krebs cycle. 6. Why does ketosis seem to help? The two main ketone bodies which are made during ketosis are acetoacetate and 3-beta-hydroxybutyrate, and these can be converted into the short-chain fatty acids acetic acid and butyric acid respectively. Okay, let's talk protocols. There are three fundamental aspects to a protocol based on this new model. 1. Raise short-chain fatty acid levels in the body. This lets the feral tissue know that its services are no longer needed and it can become benign tissue again. 2. Clean any viruses, mycotoxins, parasite metabolites, and chemicals off the hydrocarbon receptors. Temporarily seal the receptors so the toxic ligands can't reattach to them, and detoxify the toxic ligands while they're free floating and thus vulnerable to being neutralized. 3. Raise the extracellular pH of feral tissue, that is, make it more alkaline so that migration of individual feral cells is less likely. From what I have learned, I believe that any protocol for feral tissue must include these three steps: raising short-chain fatty acid levels, detoxifying the receptors, raising extracellular pH. As long as these three fundamental steps are taken, additional protocols can vary based on the experience and preference of the practitioner. Okay, let's get into some more detail about how to implement these protocols. How do we raise short-chain fatty acids? First, measure and monitor stool pH. This is an indication of short-chain fatty acid production. For more information on how this is done and what the readings mean, I suggest you watch our Philomet video. While the long-term goal is to have the client's microbiome supply all the short-chain fatty acids they need, this may take some time to accomplish. So in the immediate, consider supplying short-chain fatty acids in supplemental form. You can consider both rectal and oral administration of short-chain fatty acids. The exact ratios, concentrations, pH, and time-released aspects of a short-chain formulation are extremely important. I've seen some well-meaning compounding pharmacies offering short-chain fatty acids in ways I know from personal experimentation can be very problematic. You may wish to consider our Philomet and Lactamet short-chain fatty acid products. 2. How do we detoxify the hydrocarbon receptors? Viruses, mold toxins, parasite metabolites, and chemical toxic ligands can be very difficult to remove. As an example, the chemical dioxin is so firmly bound to the hydrocarbon receptor that it has a 5-day half-life in the cell and a 7-year half-life in the body. To remove toxic ligands, there is a plant called Indigo naturalis. I've discovered that certain extracts from this plant have an even stronger binding power to the hydrocarbon receptors than dioxin does. As a result, it may displace toxic ligands as well as seal the receptors while we work on detoxifying the now unattached toxic ligands. If you are interested in Indigo naturalis, we have it along with other elements that support toxic ligand detoxification in our Receptor product. 3. How do we raise pH? Research has shown that low pH is one of the main triggers that transforms benign growths into feral cells. Studies have shown that oral sodium bicarbonate selectively increases the pH of extracellular feral tissue. Fortunately, even long-term bicarbonate therapy does not affect blood chemistries or lead to systemic metabolic alkalosis. Here we have to be artful. The detoxifying of the hydrocarbon receptors with indigo extracts may be more efficient at lower pH values, that is, an acidic environment. So consider 5 days of hydrocarbon receptor detox, and then continue, but also add in sodium and potassium bicarbonate in an 11:1 ratio taken first thing in the morning. This gives us a head start at clearing the toxic hydrocarbon receptor ligands, but does not lose sight of the importance of raising pH. Consider starting with a 1/4 teaspoon of a bicarbonate mix in a glass of water and work up until the pH of the urine is 7.0. Do not take more than 6 grams in a 24-hour period and consult with your physician to see if this level of sodium is safe for you. Finally, stay hopeful. I believe that most feral tissue is not our adversary. While there may be a small percent that cannot return to a healthy and benign state, our immune system, which can be supported with other protocols, may be able to deal with these few remaining cells. In this photograph, you can see feral cells in the intestine. The pink cells in the center are the feral cells, and the blue tissue surrounding it is normal tissue. This person was treated with indigo extract in order to help visualize the border between the feral tissue and the normal tissue. The question is, why did the normal tissue turn blue and the feral tissue stay pink? The reason is the feral tissue wanted the indigo extract. It pulled it inside of itself, whereas the normal tissue had no need of it and it stayed outside the tissue. If feral tissue was adversarial, it would reject this extract. Instead, it pulls it in. It's working with you to pull in this indigo extract. I believe the feral tissue wants to recover, wants to become benign. Thank you for taking the time to listen. I'm grateful to all the researchers who published the discoveries that I relied upon in coming to these understandings, and I hope that some researchers and clinicians listening to this now are inspired to follow up on this theory with experimental and clinical work. It's a simple theory, and God willing, a correct one. If you have any questions, feel free to contact us by email or by phone. Take care.